that happen. This is an exciting semester for me. There's 4 of my colleagues, whether my lab is hosting them or not, are coming up or down to Moss Landing to talk about fun stuff. I don't know how much seaweed stuff we'll hear about today, but a photo. The charismatic algae. But it's really fun talking about, or being able to introduce Brian. When I was here as a grad student in the early 90s, Brian was a grad student across the pond over at Hopkins with Mark Denny. He had been an undergrad at Stanford and migrated over to Hopkins to do his PhD. At that time, words like engineering, biomechanics, acceleration, crazy words were coming out of these guys' mouths, talking about seaweeds that morph and change and go with the flow or don't, all really cool stuff. And I know that's where a lot got started. It was a lot of very determined work on the intertidal in some conditions that were relatively unique. Brian, after getting his PhD, went down to Santa Barbara and did a postdoc with 3 individuals that we all probably do know. Leib Washburn was one, who's an oceanographer. Steve Gaines, who's an ecologist, a marine ecologist, and then Dan Reed, who's a kelp ecologist. And so he was kind of shared amongst the 3 brains, and they did lots of stuff coming out of there. This is before Santa Barbara got that giant bolus of annual money to fund the LTER program, which has been great and highly productive. But so Brian was kind of the proto-LTER group, and I know there was a lot of stuff coming out during that time. A lot of stuff with dispersal and kelps, a lot more of the physical biological interactions. Then he got a job. And he ended up at Bodega, and I know he's got, he's been there for a while now. It's been 2004, I think, when you got there, right? And things have migrated to larger topical issues of broader scale in both space and time, which I think probably reflect on your diverse interests, which we saw a long time ago, both in physical biological interactions. And like I said, he's moved beyond seaweeds. And is going to let us know a little bit more about that. But it's great having him back down here. It's been a while. It's like you were saying, it's been 27 years-ish. 26 years since he was last here, and we were in the trailers. So there's a lot to talk about. So anyways, want to welcome Brian down from UC Davis and Bodega Marine Lab, the physical side of nearshore ecology from pH and warming to ocean turbulence.
Great, thank you, Mike, for that introduction. And thank you all for coming today. It's a pleasure to be here. As Mike pointed out, it's been quite a long time, so it's nice to come back. So what I thought I would do today is to talk about some of the work that we've been doing that kind of sits at the intersection of physical and ecological processes. The work that I'll talk about has been led by a variety of people in the lab. And what I thought I would do is to focus on 3 of those projects that those students have done such a wonderful job leading. I've kind of organized those research veins that really have been going for a while now in the lab. These are projects that have been going for a number of years. The first one is ocean acidification, and here we'll talk about some work that we've been doing to try to understand how ocean acidification influences species interactions. Then we'll turn to another physical process or set of physical processes, heat and mass exchange. Here we're going to talk about heat transfer into mussel beds and the implications that that process has for the many organisms that live within the interstices of the bed. That's kind of a low tide perspective. But then other things happen at high tide. Here we're concerned with fluid exchange to the interior of a mussel bed and potential chemical stresses that can arise on those inhabitants of beds at high tide. And then we'll close with a discussion of, brief discussion of nearshore turbulence and its implications for larval settlement. So let's go ahead and dive into the ocean acidification. This is a global phenomenon, a global process that's probably familiar to just about everyone here. But it's what happens when the carbon dioxide that we release as humans into the atmosphere also enters the ocean. About 30% of that CO2 enters the ocean, and it drives a series of reactions that Scott Doney has shown in this nice diagram here. Again, many of you are familiar with this, but let me just go through it quickly for those of you who may not have been thinking about it recently. CO2 enters the ocean. It combines with water, it forms carbonic acid. The vast majority of that material, that substance, dissociates into bicarbonate ions and hydrogen ions. And then a non-trivial fraction of the bicarbonate ions in turn dissociates into carbonate ions and hydrogen and more hydrogen ions. We typically think about reactions if we load them from one side, that'll drive the series of reactions to the right. That's almost correct for ocean acidification. The only difference is that notice that two of these interior reactions produce hydrogen ions. So this final reaction here actually goes the other direction. The net consequence of CO2 entering the ocean is therefore a modest increase in CO2 concentrations, a pretty dramatic increase in bicarbonate concentrations, increases in hydrogen ions. That's what leads to the phrase ocean acidification, because there's a lower pH that arises, and then a decline in the carbonate ion concentrations as well. And it's these latter two effects that people are most concerned about, because the reduction in carbonate ion concentrations, that's a building block for calcium carbonate, the material that makes up the shells and the skeletons and the tests and the spines of calcifying organisms. So that process of calcification becomes harder. But the increase in hydrogen ion concentrations has separate implications, because marine organisms have to regulate their acid-base conditions, and so that can become more challenging in the face of ocean acidification. So this is one of these big topics now, and there's literally hundreds of papers coming out a year now. And some people might say that that's a fad, and it may be, but it's an important fad, I think. Lots of papers. Most of those papers, there's actually, to take a step back, there's actually a lot of attention now on multiple response metrics, so multiple consequences of ocean acidification, consequences for growth, survival, reproduction, calcification, and so on. There's also increasing attention being paid to the fact that ocean acidification may not occur in isolation. It accompanies things like reductions in oxygen or warming temperatures, and there's people here that are studying those kinds of multiple stressors. But one really important accompanying point is that the majority of studies still, even today after hundreds and hundreds of papers, still target single species, usually in a lab setting, operating in isolation. And of course what that means is that there's a minority of studies that actually are addressing species interactions, and that's a gap. That's a gap because species interactions can be critical. We know from the terrestrial climate change literature that many of the most dramatic, abrupt, most apparent changes that arise from climate change arise not because of the direct effect of the climatic driver on a species, but because one species is influenced by that climatic driver in a way that changes how it interfaces with another species, and it's that species interaction that then causes a flip or a transition or a phase change in the ecosystem. That's not particularly a surprise, because if we think about ecology as a field, species interactions are central to ecology. We always talk about predation and competition, facilitation and parasitism. These are the main themes in ecology. And just a couple, actually it was more than a couple years ago, a few years ago, we wrote a review paper where we kind of went through some of those connections between ecological theory and the potential places where ocean acidification could impinge on these key principles. Okay. So species interactions are important. They're understudied relative to other kinds of work in ocean acidification. If you're interested in species interactions involving animals, you're most likely going to impinge on behavior. And early research, in fact, showed that there can be detrimental effects of ocean acidification on behavior, on sensing and cognition in fish. Some of this is probably familiar to a number of you here, given lots of fish work that happens here. Those effects, those consequences for sensing and cognition were thought to arise due to the altered function of neurotransmitter receptors. And the kinds of things that were happening under ocean acidification were actually quite bizarre. that really caught people's attention. Things like larval clownfish that are swimming towards predators rather than away from them, pretty dysfunctional. A lot of that work has been done in fish, but there's now growing number of studies that are showing that invertebrates can also exhibit altered behaviors under ocean acidification as well. And so we got interested in that issue a few years ago. And in particular, we got interested in whether ocean acidification might influence the behaviors of tide pool organisms. And we really latched onto tide pool organisms because we also, in thinking about ocean acidification, kind of thought that tide pools might provide a window into future behaviors, things we might see as ocean acidification ramps up in the future. And that's because pH levels in tide pools can get tremendously low at low tide during nighttime low tides. So just to kind of walk you through some of these data, what I'm going to talk about next is work by Brittany Jellison, who finished up in my lab about a year ago. She's now at Bowdoin. Here are some pH data from very close to the shore, just off the rocks at Bodega Marine Lab. And you see that the fluctuations here are notable. The fluctuations that you see right adjacent to the shore are substantially larger than what you see out in the open ocean, where pH conditions, carbonate system conditions are quite stable. Here's a blow up of a 24-hour segment, and you see that the pH over a diurnal cycle can vary by about 0.2 units or so. That's out just along the shore. If you go into tide pools, the white bands here are low tide, and you see if those low tides occur at night, you can get extraordinarily low pH values down as low, we've measured as low as 7.0. These are very, very low values. You'll never see them out in the open ocean, even many decades hence. At low tide during daylight hours, you see the opposite thing happening. And of course, these massive fluctuations are because you have an isolated water mass that at nighttime is accumulating respiratory carbon that's released by all the organisms that are living in the pool. On the other hand, during the daytime, micrograms algae are over here photosynthesizing, sucking up that CO2 and raising the pH. So the kind of perspective that we approach these next experiments I'm going to show you with is that ocean acidification is likely to just superimpose on these really dramatic declines in pH that you see, for example, in nighttime low tides and tide pools. And that suggests then, if you kind of walk with this logic, is that invertebrate behaviors might be altered in tide pools sooner than in other habitats, kind of why we were motivated to explore these issues. There's a number of different behaviors you could consider. The obvious ones to start with are anti-predator responses, like the ones that many, many species exhibit in response to Pisaster ocraceus, the ochre sea star. So there's a lot of species that exhibit really dramatic flight responses if they detect either dissolved Q that's emitted by these animals or direct contact. And here's just a quick list of different groups of organisms that exhibit these flight responses: abalone limpets, periwinkles, whelks, turban snails, and multiple species in each of these groups. What Brittany focused on were turban snails, black turban snails. So under ambient seawater conditions, what you would see if a black turban snail were to experience Q from a Pisaster is that it would start to flee up the side of the tide pool, it would come up to the water line, it would exit the water, and it would settle itself just above the water. And if you've got many of these animals, what results is this snail halo. And the thought behind this is that, the understanding of this is that by leaving the water, that provides a spatial refuge from predation for these snails, because Pisaster, even though you can find it out of the water at low tide, that's because the water has receded. It doesn't tend to leave the water as it is searching for prey on its own. So this is thought to provide a spatial refuge, this behavior of exiting the water by the snails. That's under ambient seawater conditions. The obvious thing to ask then is what happens when the pH goes down. So what Brittany then did was some controlled laboratory experiments where she provided snails with dissolved Q from sea stars, or wow, that went all the way back to the beginning. Let's see here. Sorry about that. Yeah, that did. Let's see where we can go here. There we go. Okay, sorry about that. Not sure what button I hit there, but I'll try not to do it again. So she set up controlled laboratory experiments where she provided snails with sea star Q or not, and verified that, in fact, in the laboratory she can reproduce these behaviors where the snails exit the water when they experience Q. And then she set up 16 different levels of pH and tracked what the snails did if they were subjected to Q from sea stars at those different pH levels. And this allowed her to determine the threshold pH at which these flight responses became impaired, because they indeed became impaired, which is what this slide here shows. Black turban snails don't flee the water under low pH. So what we've got here on the y-axis is the proportion of time that these black turban snails are out of the water. On the x-axis is pH. Notice that we're going to very, very low pH values. Probably won't even get these, even in tide pools, these lowest ones, by 2100. But we might get these values over here by 2100 in tide pools. You'll notice there's both white dots and black dots. The white dots correspond to what the snails do when there is no Q, so they don't leave the water. They just stay in the water. But if there is Q, under ambient seawater, boy, having trouble with this pointer here. Under ambient seawater conditions, if the snails experience Q, they do flee the water. This is just what we talked about before. But at lower and lower pH levels, there's a decreasing tendency for the snails to leave the water, such that at pH levels of about 6.9 or 7, about 30% of the snails are spending about 30% of the time out of the water, as opposed to closer to 100. So what this means is that ocean acidification is likely to leave this very common grazer, it's familiar to anyone who walks around on the shore out there, vulnerable to predation. So that's a nice insight, but it's really just the tip of the iceberg, and it essentially begs the question as to what else might happen in these ecosystems. What else might happen in these food webs due to altered pH in future decades? And so that leads us to the next project that Brittany led, where she looked at cascading indirect effects and how ocean acidification might influence those indirect effects. And the idea here is that modified interactions between sea stars and snails might also influence macroalgae. So yes, the sea stars are eating the snails, but the snails are eating, and here's your macroalgal photo here, Graham. They're also eating the algae, and so if you change this interaction over here, it's possible that this one downstream could also be affected. And in thinking about this, there's probably multiple hypotheses we could form, but two of the more obvious ones are these. One, sort of really obvious, if the snails don't leave the water under low pH, then that places them in a location where they're likely more vulnerable to predation by sea stars. So we might expect that more of these snails will be eaten under ocean acidification by sea stars, leaving fewer of them to eat the macroalgae. And so we might predict, okay, the grazing rates on the macroalgae are going to decline in total. That's one possibility. Unfortunately, there's a completely opposite prediction one could make as well, and that is that this loss of fear, in essence, by snails, the fact that they remain in the water, might alter their foraging. They're down in the water, the macroalgae is sitting right there, close vicinity. Maybe they'll actually eat more macroalgae, even though they might have higher vulnerabilities to sea stars. So these are two potential outcomes, but they really come back to this question here. How do sea stars affect the amount of algae consumed? This is this indirect link here in the food web that's accomplished by this, by sort of a transduction of effects mediated by the snails. That's what we're after. So to get at this, Brittany conducted some mesocosm experiments. These are some elements of one of those mesocosm experiments. Here she constructed containers that had different combinations of species. So this one over here on the left, for example, had black turban snails and some little discs cut out of the blades of Mozziella. That kind of situation there provides an estimate of the baseline grazing rates accomplished by snails. This one here, she inserted a sea star on the backside of this barrier. So the Q, the dissolved Q from the sea star, is available to the snails, and so they can respond to that Q and react to it. But the sea stars can't eat the snails. Over here, they can actually eat the snails. So you can kind of see how this would work. The first two treatments, by comparing them, that allows the effect of the predator on the grazing, the effect of the predator, now remember we're talking about indirect effects, on the grazing rates via shifts in snail behavior. That allows for the assessment of that effect. The two on the right allows the effect of the predator on grazing rates via consumption of snails to be assessed. So to use the language that some people like, this is the trait-mediated indirect effect, the TMII. I'm not going to use that again, but for those of you who care about that, it's too much of a tongue twister. Talk about an inefficient word or phrase. Whereas this is the DMII, the density-mediated indirect interaction. And then, of course, she did these experiments under both ambient, seawater, and low pH, because after all, the real question that she's after is what's the effect of ocean acidification on these connections in the food web. Let's go through each of sort of multiple steps in thinking about this. The first thing that she was able to show is that both snails and sea stars are affected by pH. We already knew the first point, that snails don't flee the water under low pH, but we can look at the data. So here we've got the proportion of time the snails are out of the water under ambient pH. So when there's no predator, the snails stay in the water. They like the water. If either predator Q or the predator itself are present, the snails flee the water. That's just what we've seen before. That's under ambient conditions, and we already talked about how under low pH, even if the predator Q or the predator itself is there, the snails don't flee the water, they remain in the water. That's just what we've seen before. So she just verified the patterns that she'd seen in her previous experiment. The sea stars, they are also affected. There's a variety of metrics we could track. Here's just one of them. It's the distance traveled by the sea stars in some defined period of time. And we see that under ambient pH, they move around. They kind of move around similarly, whether there's no prey or prey Q only, or whether the prey itself is there. So there's not really evidence that the sea stars are tracking the prey. They're just kind of bumbling around, and if they bump into a prey, they consume it. However, under low pH, we see that the sea stars move less. So I think you can see, again, we're sort of in one of these ambiguous situations. The snails fail to flee the water. That puts them down in the water where the sea stars could go after them. On the other hand, the sea stars don't move around as much. Which of these might play out more strongly? It's not obvious. So I could have a show of hands, but I won't. As it turns out, the sea stars eat more snails under ocean acidification, and appreciably more. So the probability of a snail being eaten under ambient pH is low, substantially higher under low pH. So more snails are killed, but we still haven't answered that question about what happens to the macroalgae. And we're still left with these sort of competing predictions. Does less macroalgae get eaten because more snails are killed? There's fewer of them to consume the algae. Or do the surviving snails eat more macroalgae because the macroalgae is right there handy underwater with them? Well, the answer to that is that the remaining snails actually eat more macroalgae under low pH. So if you look at the macroalgae eaten under ambient pH, eat more macroalgae than, Let me just walk through this. So the amount of macroalgae that's eaten under ambient pH in the absence of predators up here. If the predator Q or the predator itself are present, again, remember the snails leave the water, so not as much macroalgae gets eaten. That's just what we would expect. Over here is the somewhat more non-intuitive one, or the one that was ambiguous. And we see that here, if the predator Q is present or the complete interaction is, the snail's right there where it can eat them. These are all similar in terms of the amount of macroalgae eaten. This is a little bit depressed relative to the other, probably because some of those snails are being consumed by the sea stars. But regardless, the effect on the macroalgae is that the effect of the predator on the macroalgae, so the strength of the indirect effect is weakened. So we see that the consequences of ocean acidification is a weaker total top-down indirect effect, another mouthful for you, of predator on basal resource. Okay, so lots going on here. The main point really is that species interactions matter and they are susceptible to ocean acidification. So we already talked about detrimental effects of ocean acidification can manifest through altered relationships among species. The work that we're doing is not unique. There's people just up the road that are doing work on species interactions under ocean acidification. Christy Croker, a former postdoc of ours, is now at Santa Cruz, is doing a lot of work on species interactions. This is not unique in terms of that conclusion, but it's certainly something we need to pay attention to. Intertidal taxa, including very common ones, exhibit behavioral sensitivity to ocean acidification. So black turban snails, sea stars, a number of limpets we've also looked at, don't have time to talk about. Many species in these intertidal systems are susceptible behaviorally to ocean acidification. And we can conclude that although the consequences of ocean acidification for trophic connections are relatively underexplored, particularly the behavioral ones, they're likely important and probably warrant further attention. Okay, so let's move on from ocean acidification. We'll come back briefly to it in a second, but talk about heat and mass exchange, and in particular thermal protection within mussel beds at low tide and chemical stresses inside mussel beds at high tide. So why mussels? Well, first of all, they're a foundation species. They competitively displace other large macrofauna. That's been known for a long time. But they also provide really important habitat in the interstices between the shells of these animals for literally hundreds of other species. And that's nicely shown in this little sketch here where you can see all kinds of things. Sea anemones, juvenile crabs, polychaetes, sea cucumbers, chitons, barnacles, limpets, about everything you can imagine down inside the interstices of these beds. That's relevant because intertidal invertebrates have been model organisms for studying consequences of climate change for quite some time. So there's been really important climate change work been done on mussels and all these other organisms. So lots is known about thermal stresses at low tide, including things about local adaptation, potential for climatization, lots of details here. But if you actually think about this and look into these papers, you'll recognize there's a mismatch here with how things often operate, in that most of the information concerns solitary animals, whereas at least the younger life stages of many of these animals live within the mussel bed. And in fact, as you might expect, the physical conditions within aggregations of these densely positioned mussels can be quite different. So this is known for a long time. The literature is rife with commentary about how beds of Middleus californianus, the California mussel, can reduce environmental stress, reduces temperatures down here, reduces desiccation stresses down here, can maybe attenuate wave shock. And again, more than 300 species take advantage of these complex structural areas inside the middle of these mussel beds. But one of the things that Laura Juergens, who finished up a couple years ago and is now an assistant professor at Texas A&M, what she pointed out to me along the way is, when you start reading the literature, you realize there's a lot of commentary about this, but there's not a lot of data. The extent of the environmental buffering really was not been quantified very well. So clearly there was lots left to learn about the performance and survival of organisms inside these beds. And as it turns out, again, no surprise, but we didn't really know the extent to which this might hold. Stress amelioration within beds can be substantial, and there's also this sort of subtext here. They can also be stress exacerbation. These are data, these are maximum monthly temperatures measured at Bodega Marine Lab, on the shore at Bodega Marine Lab. And here's data, here are data from inside the bed, and then here's some temperatures on the rock outside the bed. And then the gold inverted triangles are temperatures on the surface of the mussel bed. And what you can immediately see is there's a dramatic difference here. And in fact, the maximum temperatures can be reduced by up to 19 degrees C, just moving over a few centimeters from the top of the bed into the interior of the bed. The other thing, if you squint a little bit, what you can see is that the surface of the bed during those summer months is a little bit hotter than ambient temperatures and hotter than the surrounding rock. So there's some stress exacerbation going on if you're associating yourself, if you're an animal, associating yourself with the very surface of the bed. So these are measurements on the physical side. What does it mean? Well, the modulation of environmental conditions likely has consequences. This conclusion was possible from some laboratory experiments that Laura conducted, where she focused on juvenile mussels. These settle routinely into the interior of mussel beds. And she explored the thermal limits of different size classes of these mussels using some temperature challenge experiments. So what you see is the small ones that are sort of 4 to 6 millimeters in length. They are happy as clams. Interesting that I chose that phrase. They survive quite well up until about 30 degrees C, at which point their survivorship declines precipitously. The bigger juvenile mussels can cope with somewhat higher temperatures before they decline. And just to remind you, kind of place context here, the summertime maxima sit out here, which tells you that these small guys are let me take a step back. So that's the outside temperatures. The inside temperatures are down less than 25. So what that tells you is that the small ones are benefiting quite profoundly from being inside the bed. We predict substantially enhanced survival of small juveniles within beds. And then the other side of that coin is that, boy, I'm just sorry about that one too. If you look at the bed surface, you'll see that basically these small ones are toast if they're on the bed surface. So it's dramatically low survivorship predicted for small juveniles if they're at the surface. So they are really benefiting, based on these laboratory experiments, from these thermal amelioration effects of mussel beds. These are just lab data. I could show you plots from the field. I'm not going to do that, but they're entirely consistent with this. So small mussels that are on the surface of mussel beds, they get cooked and they die. Very good survivorship if these juvenile mussels are inside a bed. Okay, so those are data, at least the physical data, were from Bodega Marine Lab, but Laura also made measurements of temperature conditions up and down the west coast of the US, a whole bunch of different places, and this thermal buffering within mussel beds applies across those biogeographic scales. So this is a data set from Taylor Point, Washington. The blue dots are the temperatures inside mussel beds. The white triangles are outside the bed, and again you see dramatic reductions in temperature inside the bed compared to outside. Again, not a surprise, but this tells us kind of the scope of these benefits. So similarly attenuated temperature extremes in beds in Washington state, much like we saw at Bodega. And what that means, what that implies, is that the effects of habitat formers trump, that one was intentional, latitudinal trends in temperature. So this is a bit of a complicated slide, so let me walk through this. Up here we've got the number of hours per month that exceed 35 degrees C. 35 degrees C is a typical lethal thermal maximum for a lot of invertebrates. Down here, we've got temperatures, the number of hours per month that exceed 26 degrees C. That's kind of a typical threshold for sublethal stress where heat shock proteins are activated. And then we've got latitude down here. So a couple sites at 34 degrees latitude near Bodega and California, 38 degrees Northern California, somewhere in there, and then up in Washington. And what we see is that if we're out on the rocks, we exceed these thermal limits quite, I don't know if commonly, but certainly it happens. Whereas if we're in the mussel bed, it never happens. And similarly for this sublethal threshold. Lots of times when these animals are going to experience stress if they're outside the mussel bed, much lower cases when they're in the mussel bed. These are data from high shore spots. On the low shore, we get some additional algal turf coming in, so Neorotamella forms these dense canopies, has a similar effect to the mussel bed. So the temperatures outside on the rock are higher, routinely exceeding some of these thresholds, but not so within the canopies. And that kind of finding, you can step back and think about the implications of this, because it does have some repercussions for how we think about latitudinal temperature gradients. One of the big themes in ecology, any ecology textbook you pick up, will talk about the latitudinal temperature gradient and its relationship to diversity. And so there's this idea that, and it's a real pattern actually, that temperatures, average temperatures, are higher near the equator and decline as you move towards the poles. An intertidal system is a little bit more complicated because of the timing of low tide. Areas up in Washington, low tides occur in the middle of the day, so the thermal stresses might actually be higher here than a little bit closer to the equator. But in general, there's this kind of idea that there's this latitudinal trend. But that ignores the effects that Laura has documented, that if you are one of these many organisms that lives within a dense biogenic habitat, within the interstices or of a dense aggregation of organisms, that latitudinal thermal pattern disappears. So it raises the question about what is the meaning of the correlation between the latitudinal temperature gradient that holds outside of dense canopies and the patterns of biodiversity that we see. Maybe there's something there, but it's worth thinking a bit more about. Another question is this one: what happens if climate change threatens a habitat former? Remember, mussels support as many as 300 additional species. If the mussels are threatened, that could have some sort of tipping point kinds of consequences. So some food for thought generated by Laura's data. Okay, that was low tide. Let's turn now to high tide. So now we're going to transition from the processes associated with heat exchange to issues of mass exchange. We're talking about fluid mixing from exterior waters into the interior of a mussel bed. And the question here takes us back to ocean acidification. Could conditions of ocean acidification be exacerbated in the interstices of dense assemblages of submerged organisms like mussels? This is work that Aaron Ninakawa is leading in our group. So these experiments involve some flow tank manipulations or activities, I guess. What he did is he transplanted a mussel bed from Bodega Harbor, this is Middleus californianus again, into a recirculating flow tank. And then using kind of an absurd number of instruments, many of which originated with Yui, who's here. And then Aaron was able to use those instruments to quantify pH, dissolved oxygen, total alkalinity, and calcium carbonate saturation state. That's a parameter that's related to the availability of carbonate ions, so it tells us something about calcification potential. And he was able to do this as a function of vertical height in the tank, all the way from down here up through the mussel bed and out into the exterior waters, and was able to do that as a function of water velocity. These are the kinds of data he collected. It's a little bit busy slide, so let me take one panel at a time. So on the y-axis over here, we've got height away from the substratum. So the mussels are kind of in this region here below that dashed line. And over on the x-axis here, we've got the change in dissolved oxygen concentration compared to that outside the mussel bed. If you go to reasonable flow speeds, 25 centimeters a second or so, you see that the fluid mixing is rapid enough that there's no gradient. There's no effect of the mussel bed on seawater chemistry. But as you drop down to slower and slower velocities, you see this gradient comes into play, such that at near stagnant conditions, you might have a reduction at the base of the mussel bed of 25 micromoles per kilogram of oxygen. That's oxygen. Similar patterns for pH. So in terms of pH, now we've got, under pretty quiescent flow conditions, a reduction of almost 0.1 pH units relative to the exterior waters. Alkalinity declines similarly, and also this calcium carbonate saturation state parameter. So these are appreciable gradients, and they're certainly not going to apply to the open coast, where these are going to be flat lines like we see over here, But these mussels do live in estuaries, and flows in estuaries in some cases can be quite reduced. So that suggests that these chemical conditions that we see over here on the left in each of these panels might accrue in some cases. And of course, the biggest offsets are occurring down at the base of the bed. So if the animals that are associated with these mussel beds are down deep in the mussel bed, they are the ones that are going to experience these altered conditions, these potentially exacerbated ocean acidification conditions, these lower pH conditions, more so than other organisms. So in terms of just sort of highlighting maybe some take-homes here, in terms of the thermal and chemical modification within mussel beds. At low tide, mussel beds strongly influence thermal regime. Stress amelioration and stress exacerbation are both possible. The effects on residents arise differently across body size. And I think it's worth thinking about potential implications of this for understanding the role of foundation species across latitude. At high tide, these OA conditions may be appreciably exacerbated. So 0.1 change, 0.1 reduction in pH is about what we've seen in the global ocean since pre-industrial time. So this is non-trivial. So they might be worsened if you are an organism that lives inside these mussel beds at high tide. Okay, let's go on here. Try to make it through this third section talking about nearshore turbulence and its role in larval settlement. So one of the long-standing areas of inquiry that people have been thinking about for a long time is larval dispersal. Most, I guess, benthic marine species produce planktonic larvae. That larval movement is what's most responsible for population connectivity. So in many of these organisms, the adults are fairly sedentary, and so what's allowing for exchange among populations is this larval phase, allowing the genetic exchange of population and demographic exchange. At the same time, mortality is high in the plankton. That's well recognized. And we also know that larvae that don't find a suitable benthic location to settle will die. These are not organisms that can live forever in the plankton. And so what that suggests, and it's one of these things that's discussed at length in evolutionary ecology texts like this one that replaced the Larry McEdwards book, it implies strong selection for improved return to appropriate sites. So with that notion in mind, what that suggests is that what we've come to call habitats, we and others, have come to call habitat scale signposts, indicators could be useful. So people have recognized for a long time that larvae use a variety of substrate-associated cues to decide where exactly they want to settle. These are often chemical cues, something that's emitted by a conspecific or preferred food item. But if you think about those kinds of cues, they're mostly available over only pretty small spatial scales. They may not even be released into the water column, but if they are, they're apt to be diluted pretty rapidly such that they're going to be undetectable once you're an appreciable distance away from that preferred habitat. And again, that suggests that if there's some kind of larger scale indicator, a habitat scale signpost, if you will, that could enable larvae to identify their arrival into the correct neighborhood, that might be something that would be selected for. And the idea is that could allow for kind of a stepwise narrowing of search activities. So there's been a lot of attention to behavior of larvae, particularly vertical swimming of larvae, and how that might allow organisms to exploit differential currents that would then carry them towards the shore. There's been a lot of attention on the cues that are emitted at the very fine scale, but much less on this possibility that there might be something about the surf zone, for instance, that would tell a larva that, okay, I'm near the coast, and maybe I should activate those behaviors that facilitate my downward movement to the seafloor, where I can find these fine-scale chemical cues. If that's the case, then that could help avoid the problem of larvae initiating fine-scale searches in completely the wrong area. And the picture that I carry around in my head is a larva that wants to end up on rocky substrate, but it gets carried into the shore in a region where there's a sandy beach, and if it were to swim down to the bottom or get carried down to the bottom by turbulence, which is probably more likely, but then initiate all of these fine-scale tasting and touching and grabbing and all these behaviors, it's wasting its time. It needs to be 5 kilometers up the coast where the rocky headland is. So if there's ways for larvae to use larger scale information, it could avoid them having to do that. And what we've gotten interested over the last few years is whether turbulence intensity might act as a possible habitat indicator. Turbulence intensity is very spatially in the marine realm. They're highest under breaking waves, especially on steep rocky shores where you get these big waves coming in where a lot of that energy hasn't been dissipated by friction with the bottom. They come in, they crash right on the shore. All that kinetic energy has to be dissipated through viscosity, and so the intensities of that turbulence can be quite strong. We know that larvae do respond behaviorally to turbulence that's weaker. If they're offshore, for example, in weaker turbulence, they will respond to that by doing different things in terms of their swimming behaviors. And there's been modeling to suggest that those kinds of altered swimming behaviors can influence where individuals will end up arriving at the seafloor. We sort of focus on something else. In some of our earlier work, we also showed that turbulence does something other than alter swimming. It actually sparks an early transition into competence. And this word will be familiar to many of you, but in case it's not familiar to all of you, let me just back up for a second. So what happens when larvae are released into the water column? They're released in a state where they don't yet have the developmental machinery to exploit a settlement cue. So even if you give them a perfect chemical indicator of their preferred habitat, they just can't settle because they don't have that developmental machinery. They're not mature enough. But as they age in the plankton, eventually they'll reach a time point when if you do give them a perfect cue, they can settle and they will settle. And that's a competence period. And what we were able to show, I'm not going to go through the data because it's published now, published years ago, but what we showed is that intense turbulence can cause a precocious shift from that pre-competent state to the competent state. Actually, I will show you one figure just to kind of hopefully drive the point home. So we're going to focus on that early transition to competence. And you'll see why, if it isn't already maybe clear yet, why we're focusing on that in the context of these habitat scale indicators. Okay, so this is the work where we kind of were able to understand this better. Purple sea urchin larvae transition precociously to competence in response to turbulence. The way we went about looking at this is we started with cohorts that were barely pre-competent. And in fact, we were sitting right at the transition where about 50% of the cohort, they hadn't developed quite as fast, so they were pre-competent. So in other words, if you exposed them to a perfect cue, they would not settle. And about 50% of the cohort were competent. So if you gave them a perfect cue, they did settle. Then we exposed that overall cohort to turbulence. And what we saw was that if we exposed them to strong enough turbulence, that many of those individuals that were pre-competent then became competent and were able to settle. So we saw an enhancement in the fraction settled because we were shifting the portion of the cohort that was pre-competent into that competent state such that they then responded to the cue. So that was a sort of whoa moment for us because we, and I don't know that other people, realized how plastic that transition between pre-competency and competency was. And certainly we didn't know that turbulence could have an influence on that plasticity, could interact with that plasticity. Just to relate you here, sort of, I almost pushed the wrong button again. Right in here is kind of surf zone conditions. Certainly you get more quiescent conditions, but these are not uncommon, these values here under actively breaking waves. These are big values, but we've measured them and they're not that uncommon. Okay, so kind of how are we thinking about this? We sort of picture pre-competent larvae drifting into nearshore waters. They experience more intense turbulence due to breaking waves. This is that kind of habitat scale indicator, this indication that they're getting close to shore and there's a lot of wave action. And if you like wave action, maybe that's something that you want to respond to. Turbulence then activates the larval sensory apparatus used for detection of chemical or other cues. That allows these larvae or transitions these larvae into the competent state. Now they're competent. If they get carried down to the bottom by that turbulence, they're primed. They're now ready to exploit that chemical cue. That's the true indicator that they are in the exact right spot. So the idea that we kind of hold here, and again, this is just a conjecture, sort of how we're thinking about it, is that this sort of scenario could combat the spatial disconnect between large-scale dispersal processes and the fine-scale use by larvae of substrate-associated cues. Okay, so that's sort of the conceptual framework. What we've been doing in recent months and days is to delve further into the turbulence-induced transition to competence. And just to be clear about how we've been doing this, we've been using a Taylor Couette cell. This is actually a design that Mark Denny, who's here today, developed years ago. He nicely calibrated it for us, so we didn't even have to do that. We just used the exact geometry that he used. But this device is a standard laboratory instrument people use to produce turbulence and actually study turbulence. It consists of two concentric cylinders. They rotate relative to one another. There's a gap between the cylinders. You put seawater in there and larvae in there. And if you spin those cylinders relative to one another fast enough, you shear the fluid strongly enough that you produce turbulence. And the turbulence in these devices has been well characterized. Now, you might be saying, that doesn't look a lot like a breaking wave. Fair enough. It's not a perfect analog to field conditions, but it's probably not as bad as what you think. Because if you go back to turbulence theory, what you know is that if you're at the tiny scale of larvae, a few hundred microns, the turbulence features, the features and characteristics of turbulence at that scale have basically become agnostic about what it was that produced that turbulence. So you could have any number of agents of turbulence production at the large scale, but those motions get translated from big eddies down to smaller eddies down to the tiniest scales of motion. And once you're at the tiniest scales of motion, you could have gotten there any number of ways. So once you're down in that turbulence cascade, in some sense, what it was that produced that turbulence becomes irrelevant from the perspective of the larva. Okay, so what we got interested in doing was conducting some phylogenetically balanced comparative experiments to explore this connection between turbulence responses and the preferred habitat of different species. And the reason we are interested in using a phylogenetic framework for this is it facilitates a stronger test of the functional significance of that turbulence response in larvae. It might sound a little bit vague, so let me walk you through that. So this is how these organisms, and we were looking at 6 species of sea urchin, kind of how they're related. Their outgroup is a sand dollar, Pacific sand dollar. And you'll notice that we've got pairs here. So what we did is we focused on paired contrasts, where we got closely related species in each of these 3 groups, where one of them likes more exposed habitats, lives in more exposed habitats, and one of them lives in less exposed habitats. By exposed, I'm talking about the kind of the characteristic levels of turbulence that might be representative in these habitats. And so the prediction then, and what this allows us to do is sort of control for evolutionary relationships here. So if there's a relationship between the habitat, the preferred habitat, and turbulence, we know that it's not just an artifact of how things are related evolutionarily. It's likely to be something that has some selective significance. And the prediction is, if that's the case, that there's some selective significance here, we would predict that more robust turbulence responses in species that live in hydrodynamically intense environments. So we're just going to go quickly through that comparison for each of these pairs. So the first pair, this is sort of a weak contrast example. Both of these species are from a moderately exposed type of place or places, species from minimally different habitats. In this case, they both show a turbulence response in the sense that more of them become competent and settle as the turbulence intensity goes up. But these lines are not significantly different from one another, so they're statistically indistinguishable. So that's a weak contrast. We can then turn to a better contrast. This is species pair 2. This pair includes one from a highly wave-impacted habitat. This is Colobocentrodus atratus, the shingle urchin. It looks like a limpet, and it actually acts like when it roams around on exposed rocks in Hawaii and other places in the Pacific, it gets pounded by waves, very wave exposed. This one is not as exposed, but it's pretty exposed because it lives in these same areas. It just lives a little bit deeper on the rocks. And what we see in this case is that Colobocentrodus atratus, the shingle urchin, exhibits a response to turbulence. larger numbers of them become competent and settle as turbulence intensities go up, whereas Heterosyntrotus mammillatus, this other one here, pencil urchin, that line doesn't have a slope that's significantly different from zero, so it does not exhibit a turbulence response. So the more wave-exposed one, subjected to lots of turbulence, has a turbulence response in its settlement activities. The one that is subjected to less intense turbulence doesn't. And then our third contrast is probably the strongest one in terms of the contrast. This is Strongylosyntrotus purpuratus, the purple sea urchin, and it lives in pretty wave-exposed habitats. It has a sister species called Strongylosyntrotus fragilis, the pink urchin. It's from deep water habitats that are much more quiescent in terms of their turbulence. In this case, Strongylosyntrotus purpuratus exhibits that robust turbulence response that initiated this work to begin with. Fragilis, that line is not significantly different in its slope from zero either, so it does not show a turbulence response. So this is, not the final answer, but it's intriguing. It suggests that there's some form of alignment potentially between species that have particular habitat preferences and this potentially useful tool, turbulence, or the intensity of turbulence, for selecting these habitats. So we continue to be intrigued on this and are therefore continuing to follow up on that. Okay, just to wrap up here, because time is running out. Physical ecological interactions all over the place in nature, and they dictate pattern and process. In terms of ocean acidification, there's lots of potential consequences of ocean acidification. Perturbed species interactions are going to be key, and I'm really happy that species interactions are getting more attention, not just by us, but by a number of other people. In terms of heat and mass exchange, thermal stresses at low tide can be affected strongly by biogenic habitat. Chemical conditions can be affected at high tide. And then in the case of nearshore turbulence, hydrodynamics can influence larval settlement. And there's some evidence to suggest that larvae might actually use might use turbulence as an indicator of some preferred habitat traits. Okay, and so I'll wrap up there. I just want to emphasize again the people who led all of the work that I've talked about today. So there's Brittany Jellison, Aaron Ninakawa, Laura Juergens, Jason Hoden. Actually, Aaron's name should go down here. I sort of broke these up by section. Jason Hoden and Matt Ferner are colleagues on the turbulence work. And then, of course, the funding agencies that made this possible. I'd be happy to take any questions. Yeah, Dan.
About that last section, if a species of a larva that favors a truly quiescent habitat, if turbulence is super important, wouldn't you expect maybe exposure to turbulence to inhibit competence, or more of those lines actually go downward?
Yeah, that was a prediction that we had, or one of the alternative hypotheses is that pink urchin, which lives in quite deep habitats, hundreds of meters, might actually, because you can have positive responses and negative responses. So yeah, I think it's a great idea. We haven't seen evidence that there's strong inhibition. There's sort of teasers of that, but that was something we were looking for. Absolutely, yeah. Yeah, Mark.
Do you know how the larvae sense turbulence?
Yeah, it's a great question. We don't. We think we don't think this. One possibility that we would be interested to pursue is whether there's stretch receptors. So if you look at the scales of motion, the gradients, are sort of right around the Kolmogorov scale. So they're likely to be experiencing strong shear over their body dimensions. And so if they're being sort of tweaked by shear, you could imagine that if they have something in their tissues that functioning like a stretch receptor, that might be a mechanical way to detect that. Maybe. I mean, they have staticists. So, if you're spinning, you have rotational acceleration. So, and Heidi Fuchs has done some work suggesting that they can sense waves by that mechanism. So the wave orbits that are producing the rotational movements that have some acceleration tied to them, they can apparently detect that. So maybe it's something along those lines too. Yeah.
I don't exactly know how to ask this, but if organisms or these larvae can detect turbulence, and they know they're in a place which is turbulent, and then can become competent, does competence also imply that they're more capable of getting out of turbulence if they don't want to be in turbulent water? Another way to ask the question is, what proportion of all the larvae that are out there get in the wrong place, get in the right place?
Yeah.
Whether they're competent or not.
Right. So there's a couple questions there.
Yeah, there are.
So it's something that we've wondered about too, so I'm going to sort of answer this indirectly, or maybe I'll get there eventually. If I don't, you can ask it again. One of the things that we've thought about is you can have fairly intense turbulence way offshore. If you've got a real windy day and you get some white capping going on, you can get pretty reasonable turbulence. Not the levels that we're exploring here, but reasonable levels of turbulence offshore. And if a larva was responding to those, that would be a false alarm, because then they're competent, they have a finite duration that they can stay competent, they might be in trouble. So that is something that we've thought about. And it's actually why we think that they respond to such intense turbulence. So the values that I'm showing you for Tom and others who are thinking about turbulence, you're looking at those values going, whoa, those are about as large as I've ever seen in the literature. It's true. So in excess of 1 watt per kilogram, that's really howling turbulence. And the reason we think that they're probably responding to such extreme values of turbulence is that the distribution of turbulence is essentially log normal, which means that for a given mean value of turbulence, you might experience really big values as well. And so to distinguish a wavy habitat, sort of going off into the ozone here, so stop me if I do too much. The only way you could distinguish if you were in a wave-swept habitat is if you are responding to the ones way out on the tail, because it's got such a long tail. And I don't think I actually got to your question. If they, yeah, you have to remind me.
Let me say it another way. Those that really do respond to turbulence and have become competent and are more capable of either moving or settling, they're probably in the minority. They're the chosen few. The majority of larvae of all these species are going to die. And so selection is really influencing what's going to happen to them. That's what I'm thinking.
Yeah, I agree with you. Yeah, I mean, you can't, these animals produce millions of offspring, right. And obviously most of those can't survive, or we'd have some problems with, space.
Select
yeah.
Okay.
Yeah, you.
So like internal waves have been demonstrated to be an important mechanism for larval transfer offshore to inshore work, so that creates turbulence, but then what you just said is it requires extreme turbulence. So can you comment about the role? Is there internal wave turbulence do you think that has any role in that, or is that just the transport mechanism?
Yeah, good question. I don't, off the top of my head, know what I'm supposed to stand over here, I think. I don't know offhand the kind of characteristic turbulence intensities that would be produced by internal wave breaking, but my sense is that they would not reach up into the levels that we would see under actively breaking surface gravity waves on the shore. So sort of different types of waves, and the ones that are really creating all that froth and.
It could be like a prolonged, instead of like acute intense versus prolonged, but lower levels of.
Yeah.
Because if you're riding an internal wave, you're constantly at a higher turbulent level.
Yeah. No, you're getting at something that we've been interested in is, there's different ways to impose turbulence. You could impose a lower level for longer, as you're getting at, or you can impose a high one for briefer. You can impose a strong turbulence and then stop it and see what happens. We haven't done all those things. We have done the last thing because we were interested in whether or not that transition to competence results in an effectively permanent shift or not, or do they regress is basically the question. And the answer to that is they don't appear to regress. So they hold that early response, how do I say this? They hold that competence that they've arrived at early for at least 24 hours. And since we're sort of talking about, at least with the Dendraster larvae that we studied, 24 hours is like 10% of their pelagic larval duration or something like that, or a day or two or something like that. Yeah, Scott.
So for the mussel bed work, have you guys done anything looking at kind of how the density of the mussel bed affects the ability for the temperature amelioration, and just sort of thinking about the latitudinal pattern now? Mussel beds are much denser up north. And
Yeah, they're
so
dense.
at what point does it?
Yeah, they're
have
denser
that?
and deeper. Yeah, we haven't. You know, Laura could probably comment better on the observed differences in the beds from site to site. She was, you know, spent many dozens of hours at each of those places. It's a great question. I don't have a good answer. You would expect it as the density goes down, the amelioration would drop. But my guess is you'd have to get to pretty low densities to see it drop profoundly. It's a guess. Yeah.
Can you speculate as to what a physiological response would be, or explanation would be for why snails would spend basically more time in more dangerous conditions, I guess, or they wouldn't haul out of the water as long?
Yeah. So,
Yeah.
I don't think it's selective. I think it's an impaired. It's an impaired response. So ocean acidification is interrupting a normal behavior that is adaptive in some way, and it's creating problems. So I don't think it, you know, it doesn't make sense in the, and maybe this isn't what you're asking. It doesn't make sense from the perspective of fitness. It's a, you know.
Yeah, I guess I'm wondering if you think that's like a reduction in Q sensing, or if there's
The
some
mechanism.
sensory ecology explanation for it.
Oh yeah. So people have looked into the mechanism. The leading explanation has to do with ion gradients across nerve cell membranes. And so let's see if I can get this right. So this is the GABA receptor that's thought to be the sensitive element in this. And what happens, the model at least, and my understanding is there's increasingly kind of people are thinking it's a little more complicated than this, but let me give you the simple explanation. Under ocean acidification, what happens is that organisms increase bicarbonate ions in the extracellular fluid to buffer against that. But that creates a charge imbalance because bicarbonate is negatively charged. And so in accompaniment to that, they reduce the chloride ion concentrations in the extracellular fluid. And GABA receptors open channels that are responding to chloride gradients. And so what happens under ocean acidification is that you end up reversing the chloride gradient across the nerve membrane, such that rather than having higher chloride outside and lower chloride inside the cell, now you've got higher chloride in the cell and lower chloride outside. So now when you open those channels in response to that neurotransmitter, you depolarize the nerve cell, which then causes an excitatory response, excuse me, rather than hyperpolarizing the neuron and creating an inhibitory response. So you can immediately see why that might screw some things up, because that neuron is doing exactly the opposite thing of what it has been evolved to do. Yeah, sorry.
If that is the mechanism, exobiology is not coming up for me, but
Me too.
if that is the mechanism, isn't that how a lot of nerve cells work for many different organisms, if not all the ones that I've learned from?
Yeah, so many of them work by mechanism of ion gradients. Not all of them are chloride.
Okay.
So there's the sodium potassium is the action potential one, and we should talk to someone who works with Gilly if we want to go too far down that. Yeah, very interesting. But there's other things. There's glutamate. There's calcium. There's a lot of ions floating around, and a lot of potential for these to get disrupted. I think the reason that people hit on this GABA receptor is that the gradient is pretty weak across the membrane. So it didn't take a lot of tweaking to change the direction at which it's going. Sodium, potassium, really big differences. And so you'd have to really change things outside to get that one to reverse. Yeah.
And so just to clarify what you're saying about that, like there's, his point kind of is that there's two different places that it could occur. It could be the Q that's being meddled with by like the chemistry around the snail versus like the snail's own biology being what's inhibited. And so your point is that it looks like it's really like in the organism that the problem's occurring.
Well, we certainly know that's the case. You're absolutely right. that if you change the pH, there's a lot of products that have charge associated with them. So if you're changing the hydrogen ion concentration, you're going to mess with that charge. In the case of the black turban snails, I didn't talk about it, but one other part of Brittany's experiment was explicitly looking at that, asking, do they not detect that the sea star is there? In the case of the black turban snails, they do, because what they do is, it's not that they don't respond at all. They will actually initiate a flight response. They just don't complete it. They don't leave the water. So it's really interesting in that sense. People are also concerned about loss of muscle function, because apparently if you change the pH enough, you can degrade muscle function. And actually, this reminds me, Haldane used to drink hydrochloric acid because he was interested in how it would influence muscle function. A little different situation than exterior waters, but it sort of makes the point that other things can be affected too. Whether that's going on with the sea stars that moved less, I don't know.
Okay.
But yeah, lots of, you start messing with chemistry, a lot of potential outcomes. But the Q degradation or not degradation under low pH is something that people are thinking about too. Yeah. We don't think that's what's going on with the sea stars, but it might go on with something else.
All right, let's thank Brian for an excellent seminar. I'm sure he's happy to answer more questions if you have.